Good afternoon, everyone. My name is Xu Lu from West China Hospital of Sichuan University. Today, my topic is about the meta-analytic imaging findings in schizophrenia and a data-driven approach to subtype schizophrenia patients. I have no conflict of interest to declare. As we know, schizophrenia is a chronic severe mental disorder with high relapse, high suicide, and high disability. Patients cannot tell what's real from what's imagined. And we know genetic and environment factors involved in the schizophrenia, but the etiology of the schizophrenia remains unknown. Because of that, there is lack of objective biomarkers for clinical work. As a famous psychiatrist said, psychiatrists remain the only medical specialist that never look at the organ they treat. For the last 20 to 30 years, radiologists have been searching for the biomarkers of schizophrenia using various kinds of imaging methods, including structure and functional methods, especially using MRI. Here, I want to summarize the meta-analytic findings of structure and functional MRI, especially in high-risk subjects, first episode schizophrenia, and chronic patients. For convenience, I mapped the findings of previous meta-analysis on the glass brain map. As shown in this picture, the blue dot means the decreased matters, while the red one means an increased parameters. As shown in these pictures, the studies of the relatives of schizophrenia exhibit either increased or decreased gray matter volume in various of brain regions, especially the decreased gray matter volume, especially in the prefrontal temporal regions, but increased volume in the basal ganglion area. When we look at the gray matter changes in first episode schizophrenia, they also showed widespread gray matter changes involving the basal ganglion, thalamus, and the cortical circuit. Here, the first episode schizophrenia is defined as the earliest duration is less than 2 years. And these findings is consistent with the changes in the relatives of schizophrenia, but they are more wide and more severe. However, when we take a look at the chronic schizophrenia patients, they showed more decreased gray matter volumes involving the prefrontal temporal and the basal ganglion areas. Here, the chronic patients is defined as the mean duration of illness more than 5 years. When we put the figures together, we can get two messages. First, the changes of schizophrenia along the illness of the course is progressive, which is support the neuroprogressive hypothesis of schizophrenia. Second, we found the consistent change in the cortical, striatal, and thalamic loop, which might be promising quantitative imaging markers for schizophrenia. When we move to the functional MRI study of schizophrenia, the findings is different from structure studies. As shown in this picture, the high-risk people showed increased activation in the right dorsal anterior prefrontal cortex, which is the most consistent findings, and the other findings varied across the different studies. When we look at the first episode schizophrenia, they demonstrated decreased activation in the bilateral parietal and the medial prefrontal cortex, while increased activation in the bilateral putamen, superior temporal gyrus, and occipital gyrus. Especially the decreased activation of the medial prefrontal cortex is the most consistent findings in first episode schizophrenia. It is interesting to look at the change of chronic schizophrenia which also demonstrated widespread increased and decreased function involving different brain regions, especially the decreased activation in bilateral postcentral gyrus, left precuneus and right occipital gyrus, while increased activations in the bilateral inferior frontal gyrus, bilateral superior frontal gyrus, left amygdala, left inferior temporal gyrus, and right anterior cingulate cortex and insula. When we put these functional findings together, we also can see a progressive change from the relatives to first episode schizophrenia and chronic patients. However, these findings are more widespread than structural findings, and involving the default mode network, sensor mode network, visual network, and silence network. Thus, the functional findings may be more state-related. Taking the structure and functional findings together, the gray matter changes have been most robust within the thalamocortic network, while the altered brain activation have been most pronounced in frontal, parietal, and default mode network. As we summarized in our previous review, these findings indicate that regional anatomic and functional brain abnormalities are significantly dissociated during the early course of the schizophrenia. One possible explanation is that the functional MRI may reflect the physiological changes related to the state, while the changes in brain anatomy reflect more stable and long-standing alterations. Taken together, there are complex cerebral abnormalities in schizophrenia. For structural studies, they found consistent findings within the biochemical cascade for schizophrenia, and the structural markers may be more suitable for subtyping the patients. While for functional studies, they found more widespread changes across the whole brain, which suggests they may be state-related findings of schizophrenia. But the relationship between the structure and functional findings remains unclear. The main challenging of the previous study is the inconsistent findings across different studies. Even in the meta-analysis, the small samples and several clinically relevant heterogeneity, including the genetic, the clinical symptoms, the cause of illness, and the treatment outcome, may confound the findings. One of the solutions to solve the problem is the subgroup the patients based on the clinic information or using data-driven approach. Many previous studies subgroup the patients based on the clinical symptoms. For example, our previous studies compare the patients with prominent negative symptoms with those without prominent negative symptoms, and the findings showing that the patients with prominent negative symptoms show more widespread gray matter changes. However, such type of studies based on the hypothesis that the patients with similar clinical symptoms have similar gray matter changes. However, that is not true. In 2015, our group first used clustering analysis to study the white matter features of the schizophrenia. In the large sample of first episode schizophrenia, we found there are two different subgroups with different white matter changes. However, they showed quite similar clinical symptoms, except the group 1 with widespread white matter changes showed more severe negative symptoms. These findings suggest the possibility that similar clinical symptoms may have different brain changes. Following that study, there are many studies using clustering analysis to subgroup the patients. For examples, we found 3 different subgroups of patients defined by the distinct patterns of regional cortical and subcortical morphological features. A similar three subgroup patterns was also identified in the independent datasets of patients from a multi-site BSNP studies. The patients showed quite similar clinical symptoms, but with quite different grammatical changes. For examples, the group 1 showed quite severe grammatical deficits, while the group 3 showed increased grammatical volumes. However, these patients showed different cognitive changes. The patients with more severe grammatical changes showed more severe Beck's scores. These findings suggest that the patients with similar clinical changes may have quite different grammatical changes. Similar findings were also identified in non-institutionalized patients with schizophrenia, and those with more pronounced subcortical changes showed more deficits of cognitive function, but with similar changes of the clinical symptoms with the other subgroups. To explore the possible pathogenesis of these imaging subgroups, we also used data-driven approach to subtype the patients based on the other features of the patients. For examples, we explore the inflammation status of the patients, and we subtype the patients based on the DNA methylation and the T receptors. And the interesting findings is we found two subgroups of the patients using data-driven approach with these methylation and T receptor features, and we found that there are two subgroups. The groups with the abnormalities of the immune system showed more severe grammatical changes, and this is quite consistent with the group 1 type, the type with severe cognitive deficits. Most of the previous clustering analyses are using the structural features. For functional MRI study, the clustering analysis is still at the exploratory stage, where further validation and increased sample size are needed. However, these are quite difficult because functional MRI to date, its no signal-to-noise ratio and elevated monitoring cost make it a poor clinical alternative for studies. And if we want to using fMRI to study the subgroups of patients, a multi-site data collection with strong effort are needed in the future. However, the initial study using functional MRI also found different subgroups within the schizophrenia patients. For example, based on the 8 cognitive dimensions, using fMRI, they found 4 subgroups, and the group 4 showed more severe brain deficits of the frontal parietal regions, showed more severe working memory deficit. Another study using functional MRI also showed distinct patterns of neural recruitment during working memory. Taken together, data-driven approaches may be used to remove the heterogeneity of the imaging study of the schizophrenia, and the structural MRI-based subtyping study is more reliable than functional study, while the functional study is at the initial stage. Finally, I want to thank all the group members and our cooperators for the study. Thank you for your attention, and welcome to China and welcome to Chengdu. Chengdu is a nice city with the name of the paradise and also is the hometown of Panda.